Hello, in today's video, we shall learn in detail about plant tissues. By the end of this video, you will have a clear understanding about the different types of plant tissues, namely meristematic tissues and permanent tissues, their structures, functions, and also differences between them. Let us start with the introduction to plant tissues. Tissues are a group of cells similar in structure and function and have a common origin. Plant tissues are primarily classified into two groups, meristematic tissues and permanent tissues. Cells of meristematic tissues are capable of dividing continuously to produce new cells. They are present only at the growing regions such as shoot tip, root tip, and at the base of internodes and leaves. Cells of permanent tissues do not have the power of division. They differentiate and attain a permanent shape suitable for their function. Now, we shall learn about the different types of plant tissues. Plant tissues are primarily classified into two groups, meristematic tissue and permanent tissue. Depending on the region where they are present, meristematic tissues are classified into apical meristem, intercalary meristem, and lateral meristem. Vascular cambium and cork cambium are the examples of lateral meristem. Permanent tissues may be simple or complex. Tissues which are made up of only one type of cells are called simple tissues. Examples are parenchyma, collenchyma, and sclerenchyma. Arenchyma and chlorenchyma are modifications of parenchyma tissue. Fibers and sclerites are the two main types of sclerenchyma cells. Complex tissues are xylem, phloem, and secretory tissues. They are made up of more than one type of cells having a common origin and working together as a unit to perform a common function. Xylem and phloem are mainly concerned with the transportation of food, mineral nutrients, and organic solutes. These conducting tissues are together known as vascular tissue or conducting tissue, which constitute a vascular bundle. Xylem consists of tracheids, vessels, xylem parenchyma, and xylem fibers. Phloem is composed of sieve tubes, companion cells, phloem parenchyma, and phloem fibers. Secretory tissues have cells responsible for secretion or excretion of materials. They are of two types, latissiferous tissue and glandular tissue. Latissiferous tissue can further be classified into latex vessels and latex cells. Glandular tissue may have internal or external glands. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now, let us look into the salient features of meristematic tissues. The cells of meristematic tissue are similar in structure and have thin cell wall made up of cellulose. The tissue consists of actively dividing cells. They are present in growing regions of plants. They are compactly arranged without intercellular spaces between them. Cells of this tissue have thin cell wall, dense cytoplasm, and prominent nuclei. Vacuoles are generally absent. Main function of meristematic tissue is to form continuously new cells for increasing cell number, length, and the girth of the plant. Meristematic tissues are classified as apical meristem, intercalary meristem, and lateral meristem. Apical meristem is present at the tip of stem and root. These cells are responsible for increasing the length of the plant. This vertical growth is also known as primary growth. Intercalary meristem is found at the leaf base, above the nodes or below the nodes, and is also responsible for increasing the length of the plant. Lateral meristems are responsible for secondary growth in plants. Secondary growth is horizontal growth and brings about an increase in girth of plants. Vascular cambium and cork cambium are the examples of lateral meristem. Vascular cambium is found in vascular bundle, while cork cambium is found underneath the bark of trees. Next, let us learn about the various types of simple permanent tissue. First, parenchyma. It is the basic or fundamental tissue found in plants. It is the most simple and unspecialized primitive tissue. It mainly consists of thin-walled cells, circular or polygonal in shape. Parenchyma forms the base packing tissue of plant body. The cell wall is made up of cellulose or calcium pectate. They are living and each cell has a prominent nucleus and vacuolate cytoplasm. Intercellular spaces are present between the cells of this tissue. Sometimes, parenchyma cell contains chlorophyll and perform photosynthesis. This kind of parenchyma is known as chlorenchyma. In aquatic plants, parenchyma contains air spaces in between them. Such a parenchyma tissue is known as aerenchyma. Next, let us study the functions of parenchyma. The main function of parenchymatous tissue is assimilation and storage of reserve food materials. They provide temporary support by retaining the turgid condition of cells. In aquatic plants, aerenchyma gives buoyancy and also helps in gaseous exchange. Chlorenchyma contains chloroplast, prepare food through photosynthesis. Parenchymatous tissues serve as packing tissues in between other tissues. They also store waste products such as gums, resins, tannins, etc. Next type of simple permanent tissue are the chlorenchyma. Chlorenchyma is a living tissue with elongated and thickened cells having cellulose and pectin at the corners. The tissue provides flexibility to soft parts of the plant so that they can bend without breaking. The cells are closely packed without any intercellular spaces. The main functions of chlorenchyma are to provide mechanical support, tensile strength, elasticity, and flexibility to stem, leaf stalks, and leaves. Next are the sclerenchyma cells. Sclerenchyma consists of thick-walled dead cells. The cells are usually long, narrow, and pointed at both ends. Due to deposition of lignin, their walls are highly thickened, due to which the cell lumen becomes very narrow. The cells are closely packed without any intercellular spaces. They are usually provided with simple pits. Sclerenchyma are of two main types, fibers and sclerites. Fibers are long, elongated, sclerenchymatous cells with pointed ends. They are found in association with tissues in roots, stems, leaves, and fruits. They are much longer than wider, with very tiny cavity in the center of the cell. Example of fibers are jute or Corchorus, Indian hemp or Crotalaria. Sometimes the sclerenchyma cells are grouped within other tissues in specific zones or regions. These are known as stone or sclerotic cells. They are very thick-walled, strongly lignified, occur in hard seeds, nuts, and stony fruits. The flesh of pear is gritty because of the presence of stone cells in it. The sclerenchyma cells are dead cells, and their main function is to provide strength and rigidity to the plant. Next, we shall learn about complex permanent tissue. The important complex tissues in vascular plants are xylem and phloem. Xylem is the chief conducting tissue of vascular plants, responsible for the transport of water and dissolved minerals from roots to all other parts of the plant. Xylem consists of four kinds of cells, namely tracheids, vessels, xylem parenchyma, and xylem fibers. Tracheids are elongated, dead cells with its both ends tapering. The walls are thick, hard, and lignified. The cell wall possesses pits. Vessels are the most important element of xylem because they are responsible for the upward movement of water and minerals. They are dead cells placed one upon the other, and their transverse walls are dissolved. They form long tube-like structures for conduction of water and minerals. Tracheids and vessels together are known as tracheary elements. Xylem parenchyma are parenchymatous, thin-walled living cells. They help in lateral conduction of organic solutes and storage of food reserves. Xylem fibers are lignified, dead cells which provide mechanical strength to the plant body. Next are the phloem. It is the chief food conducting tissue of vascular plants. Phloem consists of four elements: sieve tubes, companion cells, phloem parenchyma, and phloem fibers. Sieve tubes are the main conducting part of the phloem. It is formed of elongated, cylindrical cells arranged in vertical rows. The end walls of the sieve tube are perforated with numerous pores and is known as sieve plate. Cytoplasm is in the form of thin lining enclosing a big central vacuole. The nucleus, mitochondria, and other organelles are absent. Companion cells are living cells and are seen associated with the sieve tubes. They have a thin cellulose wall and active protoplast. Nucleus, mitochondria, and other organelles are present. The common wall between sieve tube and companion cells show the presence of pits. Phloem parenchyma are living parenchymatous cells which store food. Phloem also contains phloem fibers which provide mechanical support. They are also known as bast fibers. Another complex tissue are the secretory tissue. The cells or tissues concerned in secretion or excretion of materials constitute secretory tissue. They are of two main types: latissiferous tissue and glandular tissue. Latissiferous tissue is composed of thin-walled, elongated, branched, and multinucleate tube-like structures which contain a milky juice known as latex. The latissiferous ducts may act as food storage organs or as reservoirs of waste products. Latissiferous tissue are of two types: latex vessels and latex cells. Latex vessels are rows or more or less parallel ducts connected with one another by the fusion of their branches, forming a network. Hence, they are also called articulated laticifers. They are found in Poppy family. Latex cells are single or independent units. They branch profusely through the parenchymatous tissue of the plant. They do not anastomose, that is, they do not fuse together to form a network. Hence, they are also called simple or non-articulated laticifers. Latex cells are found in Calotropis, Euphorbia, Ficus, etc. Next are the glandular tissue. This tissue is made of glands which are special structures. These glands have the capacity to secrete or excrete products. The cells are thin-walled and the cell walls are made up of cellulose. Glands may consist of single isolated cells or small groups of cells. The glands may be internal or external. Internal glands are oil glands, mucilate secreting glands, digestive glands, etc. External glands are glandular hairs secreting gummy substances, glandular hairs secreting irritating poisonous substances, honey glands or nectaries, etc. Let us look into the differences between meristematic tissue and permanent tissue. Cells of meristematic tissue have the capacity to divide and produce new cells. Permanent tissue cells do not have the capacity to divide and produce new cells. Meristematic tissues are composed of undifferentiated cells. Permanent tissues are composed of differentiated cells. Cells of meristematic tissue are always living. Cells of permanent tissue may be either living, example parenchyma, or non-living, example sclerenchyma. Meristematic tissues are always simple tissue composed of only one type of cell. Permanent tissues may be simple or complex. Cells of meristematic tissue have dense cytoplasm and a large centrally placed nucleus. Cells of permanent tissue have a large central vacuole and normal nucleus. Meristematic tissue cells show high rates of metabolism. Cells of permanent tissue show very less or no metabolism. Cells of meristematic tissue are compactly arranged with no intercellular spaces and lack vacuole. Cells of permanent tissue are loosely arranged and have intercellular spaces. Meristematic tissues are located at specific regions of the plant. Permanent tissue cells are distributed throughout the plant body. Meristematic tissue cells have cell walls made up of cellulose. Cell walls of permanent tissue have cellulose, lignin, pectin, or suberin. Meristematic tissue produce permanent tissues. Permanent tissues are derived from meristematic tissue. Function of meristematic tissue is to assist in plant growth. Photosynthesis, conduction, and mechanical support to the plant are the functions of permanent tissue. Now, we shall study the differences between parenchyma, collenchyma, and sclerenchyma. Parenchyma are thin-walled living cells. Collenchyma are also thin-walled living cells. Sclerenchyma are dead cells. Parenchyma have thin cell wall made up of cellulose. Collenchyma have uneven cell wall made up of pectin and hemicellulose. Sclerenchyma have thick, rigid cell wall made up of lignin. Intercellular spaces between the cells are present in parenchyma. Little or no intercellular space present in collenchyma. Intercellular space is absent in sclerenchyma. Parenchyma are unspecialized and living cells. Collenchyma are specialized cells and living. Sclerenchyma are specialized, matured, dead cells. Parenchyma cells are loosely packed, thickenings are absent. Collenchyma cells have thickenings at the corners. Sclerenchyma have thickened cells. Parenchymatous cells are found in soft parts of the plant. Collenchyma cells are found in petiole, leaves, young stems. Sclerenchyma cells are found in mature parts of the plant. The living cells of parenchyma assimilate and store food, help in photosynthesis and gaseous exchange. Collenchyma is the chief mechanical tissue of young parts of the plant and resist bending, provide elasticity in young dicotyledonous stem. Sclerenchyma cells provide mechanical support to plants. Now, we shall look into the differences between xylem and phloem. Xylem consists of tracheids, vessels, xylem parenchyma and xylem fibers. Phloem consists of sieve tubes, companion cells, phloem parenchyma and phloem fibers. In xylem, tracheids, vessels and xylem fibers are dead cells and only xylem parenchyma is living. In phloem, sieve tubes, companion cells and phloem parenchyma are living and only phloem fibers are dead. In xylem, conducting channels are tracheids and vessels. In phloem, conducting channels are sieve tubes. Xylem transports water and minerals from roots to other parts of the plant. Phloem transports food from leaves to other parts of the plant. Conduction in xylem occurs only in one direction, that is, only in the upward direction. Conduction may occur in both directions, upward or downward in phloem. So today we have learnt in detail about various types of tissues, their functions and important differences between them. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.